Whenever you tell me to.
Yeah, that's fine. No, I don't get one less thing to worry
Yeah.
about. You're going on first, right?
Yeah.
Just put it right there. Come in now. You're on and off right there.
Yeah.
Should be on now. Later, turn it off. So just make sure you turn it on.
Oh.
You will? All right.
Just
all right.
been teaching a 700-person course 8 times a week with this same system. So I'm used to it.
Sure, it's up to you. You can use the stick, and then the other person,
Or you could take this. I mean, it doesn't.
No, that's okay. Okay, great. Great, thank you very much. Let me take this too.
Just make it up.
Well, I have to make it up by most of the
Yeah.
Take this one. I'm taking two.
Yeah, medical microbiology. This will you posting Twitter from my.
Do you want me to continue your talk? I can't get on my laptop, which is really frustrating. Did you ever get on?
Yeah. They had a new. We have to go get a new longer.
Oh, okay. Maybe I'll just listen to you.
Yeah, but the original one that they handed out was wrong.
Oh,
So you have to go get a new sticker.
Oh, you have your own, and
Yeah, I'm also.
this is.
I'm not going to use that. I'm going to use the mouse.
Okay.
If I use anything, yeah. I'm gonna use it.
Welcome back. Okay, welcome back. Our next speaker is Jonathan Eisen. He is a professor
professor at the Genomes Center at UC Davis. Dr. Eisen is also joined between the Department of Evolution and Ecology and the Medical Microbiology and Immunology departments at UC Davis. The focus of his research is to use sequencing and computational methods to discover new processes, how new processes and new functions arise. And he holds a PhD in biology from Stanford University. He's also a member of the American Academy of Microbiology. Dr. Eisen, please. Thank you.
Great. I think that all, yeah, works. All right, thanks. I'm really glad to be here. I do think we should sequence the city, although I'd recommend we start with a small city in the Central Valley in California, maybe first, rather than this big, complicated city, you know, in the East Coast. Jane, where are you? Yeah. We can do both, I guess. So what I'm going to talk to you about today is this project that I've been coordinating in my lab called microBEnet. And this started basically in 2010 with an announcement from the Sloan Foundation that requested proposals or put out a call for someone to carry out a sort of network-type activity coordinating communication and web resources in the community related to this emerging field of microbiology of the built environment. Paula contacted me about this, and I said, I only do research projects. I don't understand this network thing. I've never done a non-research project before, although I was very heavily invested in a variety of activities that directly relate to this, what eventually became the project. So after thinking about it for a little bit, we decided to try and do it. And we created this proposal and project that eventually was funded via the Sloan Foundation as part of their program, and it's this network, Microbiology of the Built Environment Network, and I'm going to tell you about the different components of this network. There are a bunch of people at UC Davis that I will mention that are involved in this, Hal Levin at the Building Ecology Research Group, many, many collaborators that I will mention, some of them here in the audience. And I just thought after hearing the discussion on the panel, I added this slide because I think that one of the things that I thought was very interesting about this idea that the Sloan Foundation had, which was they were launching this new research initiative, but they were thinking about how people interact with each other, how to build an interdisciplinary program, how to accelerate progress in a field. And I thought this was very interesting because I've been involved in microbial-related sequencing for a very long time. I was at Tiger, the Institute for Genomic Research, for 8 years before moving to UC Davis, and I've been funded via the Moore Foundation Marine Microbiology Program and been involved in the NSF and USDA programs to sequence microbes. And these have been sort of groundbreaking programs in terms of the science, but some of these programs had limitations or areas that they could probably do better if they had a better interdisciplinary plan in advance of the program. So eventually I put together this proposal and it was funded. And this is basically a schematic outline of what we did in the first few years of the project, which is we identified 5 or 6 major topic areas that we thought if we worked on and promoted and taught people about in the program and outside the program, it would help accelerate this new area. It would help launch microbial ecology and microbiology studies in the indoor environment as opposed to the outdoor environment where it had already happened. And this is basically what I'm going to tell you about is these different activities. So I'm just going to walk through a few of these. So one area that clearly many people are certainly aware of, that's what we're doing here, that's what many other programs do, is there's definitely need for people to meet and talk about a field, to get people from different fields together, to get people to interact with each other, to plan projects, to educate each other about the language in their fields. And so one of the biggest areas of our project is also in this area. We've been organizing a series of meetings and workshops related to microbiology of the built environment, and in particular trying to figure out how our number one sort of issue was how to get people from the microbial sequencing, microbial ecology community to interact with building scientists of very different communities and how they could learn to talk to each other and educate each other about the different areas of their fields. And so we started by organizing a series of meetings and workshops, mostly associated with large other conferences. And this is sort of straightforward stuff. The main goals were things like networking and catalyzing collaborations and producing resources. We have this exterior goal, which I'll tell you about later, which has been at every meeting that we've been organizing, we've been trying to promote and teach about the benefits of sharing to try and get the field started early on to be as open and as sharing as possible. And so this is just, for example, some of the meetings, but we targeted sort of large pre-existing meetings where we could add an additional session like this meeting on indoor air or the American Society for Microbiology and use their sort of organizational operations and add a session on microbiology, the built environment, either for the building science meetings with a lot of microbiology or for the microbiology meetings with a lot of building science. And I think this worked pretty well. I think these meetings and sessions that we organized had some benefit, but in part, going back to what Paula was saying on the panel, in part in response to reviewers' comments in our revision of our proposal for the second round of funding, reviewers and many people in the community suggested that what we needed in the future was smaller targeted workshops on specific topics where you would bring people together with a specific goal to achieve, some end product. And so, for example, one that we're really interested in the future is not just studying humans in the built environment and the human microbiome interaction with the built environment, but what role do animals play in the built environment? Can we use animal systems like zoos and animal shelters, et cetera, to learn about the built environment? And the second one on this list is for Jane. Microbial eukaryotes in the built environment, which we have targeted as a really important area, a highly neglected topic. And I agree with what Jane was saying, that there's been a very heavy focus on bacteria in particular and less focus on many of the other microorganisms in the built environment. So we're going to be organizing a workshop on this topic to bring together just sort of a couple, 10 people or so to plan how to catalyze work in this area. And there are many other topics that we have identified, largely either from ourselves or from the reviews of our grant proposal, but we've also started soliciting the community for suggestions for meetings. And one of them that we're actually doing this summer, there's a building science education meeting at Biosphere 2, and a graduate student from Jessica Green's lab, she's a grad student, right, Gwyn, has helped us plan a microbial ecology session at this building science education meeting. And so we're trying to use the meeting organization not just to catalyze sort of communication, but also to engage the entire community of researchers in thinking about what the next most important topics are. I could go on and on about meetings, but most people here are probably used to the concept of organizing meetings as part of a program to try and educate and create community. And I think some of these other topics that we've been working on are probably less discussed in many communities when you're trying to launch a new field. So I'm going to spend a little bit more time on them. So another area that we've been very heavily invested in is trying to leverage digital and social media for the community to try and better build the community and spread information to practitioners, to the public, and to funding agencies, and so on. And I'm going to give you a couple of examples of this. So for example, there's a new paper that comes out in the field. People were talking earlier about, can we use Watson to scan through all of PubMed and help people scavenge the literature? And when new papers come out, everybody's overwhelmed, right? There's so many papers coming out. How do we find them? How do we identify them? So what we're trying to do as part of this project is to help the people in the field keep up with the literature. So if a new paper comes out of interest, and we have, something to say about it, we might write a blog post about the paper, pointing people to this paper, saying something about it. We might also post that to Twitter, leveraging whatever we can about social media. The blog posts get posted, something about them to Twitter and to a Facebook page. It's a sort of standard overwhelming social media activities. Just a point to people here who are not sort of using a lot of social media, I would recommend it for communicating about the field, but the key part of the word is social. So if you just post a new paper, something about it to Twitter, and then you don't engage in a discussion about it, people very rapidly ignore everything you're doing. So we've been trying to spend time figuring out how to best sort of engage people with Twitter or with Facebook or with LinkedIn or with comments on blog posts, and I'd be happy to talk to people about this. We've sort of experimented with different systems about this. Another thing that people may not be aware of, there's a new commenting function in Pubmed. If you're an author in Pubmed, you can register for an account, and then if you comment on a paper, anytime anybody does a search of Pubmed, it's listed right below that entry that there are comments on that paper. And so when we have blog posts about a paper, we now post to Pubmed saying there's a blog discussion of this paper over at this site, and it actually engages the audience much more broadly because lots of people still do Pubmed searches all the time. And I'll mention this in a little bit. We are also trying to keep track of all these new papers we find via online sort of social referencing systems, and one that we're using is something called Mendeley. So again, maybe there's a new paper that comes out and we don't think it's worthy of a blog post, but we want to tell people about it, so it just gets a Twitter post. I'm sorry if that's happened to your papers, but Twitter is a much more rapid means of communicating about them, and we may just do direct posts. But again, whenever we find a paper that seems of interest to the field, we add it to this online reference collection so that anybody in the field can then go to this site, this Mendeley site, and find all of the papers that we've been tracking related to this field. And I'll just give you an example of how easy this is, that anybody who works in this area could contribute to this. So here's a paper that came out very recently, funded in part by the Sloan Foundation. The first author is Rachel Adams, who's a graduate student at Berkeley. We saw the paper, came out in PLOS One. I have a little Mendeley browser button. If I click on that in my browser and there's any document identifier associated with that website, any DOI, it automatically uploads it to our Mendeley collection. It's a one-click basically addition to this site. So I see the paper, I add it to the Mendeley collection. It takes 10 seconds, depending on your web connection speed. And then usually I'll also post something to Twitter, might be very bland, saying I'm just reading this paper. I did this from the back of the room earlier today about Rachel's paper to get this little screen capture. Rachel is also an amazing student. She also blogs all the time about her papers, communicates with the public, and does lots of activities. So she's been blogging on our site about her papers, and a couple of the times I've seen her papers before she has. So she finds out about her papers from Twitter, me posting about her papers. For meetings like this, we also try and capture sort of all the traffic at the meeting, all the discussion, because it's everybody here might be taking notes, but if there are any people posting to Twitter or to other social media sites, we will capture this information. You can record it in this system called Storify. That can then be shared with other people, and people can find out a better record of what's going on at the meeting, sort of like old meeting reports used to be, which people don't publish quite as much anymore. So this is another way of sort of recording. So here's an example, a recent meeting we had at UC Davis called Publish or Perish. It was about the future of scholarly publishing. Allison Fish, a postdoc at UC Davis, helped organize the meeting, and we kept track of all the discussions of all the speakers, all their slides that they posted, and all the videos that were associated with their talks. We posted those all to this Storify site so that anybody, anytime, can find out what was happening at the meeting. We've been trying to figure out ways, not everybody uses social media. So we have an email newsletter that we also send out that gives a monthly roundup of what's going on in the field, basically. We have a lot of people who subscribe to this, and we get a lot of clicks from it. I detest all email, so I'm not sure whether I'm a good role model to talk about the value of having an email newsletter, but many people seem to like having this sort of monthly roundup of everything going on in this field to try and tell people what are the new papers, what are the new meetings, what are the new grants that were funded by different agencies, what are the new funding opportunities. And we've been trying to experiment with other ways to get the word out about what's going on in the field. And so, for example, Holly Bick, who was a postdoc in my lab, just took a faculty position at Birmingham, was working on this project. She would go to some meetings where people who were working on the field were attending and do little video interviews to discuss what are they doing, what is the latest work that they're doing, and these get posted to YouTube. And again, it's sort of reaching a different audience than normal social media reaches, and it's a really nice way for people who are new to the field in particular to come in and get an introduction to sort of, this was mentioned before, what do scientists actually do when they're working in these areas? And that's sort of the people behind the science purpose. We spend a lot of time trying to track news media discussions of microbiology of the built environment. It sort of comes in waves. There's a new paper that comes out and then a bunch of stories that come out. And we either post this to Twitter or we write a blog post about it. There were 2 articles actually on the same day that came out from Brooke Burrell, reporter, one for Nova, and I forget what the other, I can't quite tell what the other one was in, I can't remember. Both sort of about microbiology of the built environment. So we wrote up some blog posts about this. Another scientist in my lab, Holly Gans, wrote an extensive discussion of how pets influence the built environment. That's partly because we're really interested in this animals in the built environment area, but we're just trying to, again, serve as a vector for information about this growing field and make it as scientific as possible, but try and have it be more reachable than many of the scientific papers that are coming out in this area. And this sort of relates to this, these sort of overlap a lot. Everybody I know in the field is overwhelmed all the time with new papers, with new information, with new data, with new $1 million prizes. I mean, every 2 weeks there's a $1 million prize about something. So, and so we're just drowning. And so one thing we view as one of our main activities to try and help the people in the field or people that might be interested in the field stay afloat and keep track of what's going on and provide a filter, in essence, for this information overload for people. And I've already mentioned that we're doing this, with new publications, how to keep track of them by recording them. And I just thought I'd plug again this social referencing system. We've been using this system called Mendeley. It's not our ideal system. It's not a completely open environment. Although anybody can join, it's not open source and it's not open. We tried to use an open equivalent called Zotero, but it didn't quite have all the functions that we wanted, so we ended up sticking to Mendeley. We are transitioning over to Zotero now. So we have 950 papers that have been added to this collection. They get curated tags, so people go through in our group and Hal Levin and other people and add tags about the papers. Are they about fungi? Are they about airplanes? Are they about microbial eukaryotes? There are probably 2 papers or something. What are they about and how people can track those? Those tags are not easily searchable, but you can search them and go find all the papers about hospital microbiomes, for example. There are actually 149 people that have joined in this group, so the people, it's sort of a socially aware referencing system. So it's not just that we're adding to an EndNote library. We are communicating with other people in the field, and anybody can post, anybody can join this group. It's completely open. Anybody can add references to the group. Occasionally, if they're not relevant, I will delete them, but that's really, really rare. Most of the additions that we've had from the community have been very helpful. And having this reference collection now serves many other purposes. So we've gone through the reference collection, tabulated all of the authors in the reference collection. We've now tabulated all of the authors that have Google Scholar pages from that reference collection. So you can now go to our tabulated list of Google Scholar pages for people that work in the field. And if you like to use the interface of Google Scholar, which there are many uses of, you can go immediately and find all the people that work in this area. We've been doing a bunch of analysis that I'll show you in a minute of what journals people publish in this area and getting contact lists. So we've been adding, sending out email with an invitation to all the authors in this group saying, would you like to subscribe to our newsletter, for example. But having this sort of collection of references that are specifically known to be about the field has been very useful. So here's a list of all the journals that are in our reference collection, and if you do a simple count of the references, the number 1 journal is Indoor Air, the number 2 journal is AEM, Applied Environmental Microbiology. That we probably expected. We didn't really know what the rest of the papers would be in. So if you get rid of those 2 journals, you can see what other journals people are publishing in that seem relevant to this field, and it's actually quite a few journals that we were not paying attention to when we were scanning journal space for what journals might be interesting. So this has influenced greatly what we do to try and screen papers to then share with other people in the community. Again, here's the Google Scholar list, and I just, if people don't know this, there's a really useful tool that you can use in Google Scholar if people have a page, and again, many of the people in this field have a page. You can go to their author page. This is a page for Maria Dominguez-Bello, who does really interesting work on the human microbiome and a variety of ecosystems. And you can follow new articles by this author. And whenever a new article is published by her, since she has a Google Scholar page, Google Scholar regularly scans through new publications, and if it matches what it appears to be her sort of cluster of publications, it gets added to her publication list automatically. And then I get an email about that. So I have this for everybody who publishes in the field. So I get announcements from Google Scholar whenever new papers come out. That's how I knew that Rachel Adams had new papers before she did. And it's, again, leveraging the existence of Google Scholar in a way to keep track with what's going on in the field. And this was discussed a little bit earlier. One thing that we would like to do but have not yet is take the full text of all of these papers and start to use language processing, clustering algorithms like Texpresso or other algorithms to try and understand in a little more detail about what people are doing in the field. What are the organisms that interact with what other organisms? Just like people have done protein-protein interaction networks, which was mentioned, we can also potentially do organism-organism interaction networks or organism-environment interaction networks. We're working also trying to do co-authorship networks. These are sort of things that we have planned in the future. By having this database, we will be able to do these activities, although we're not experts in language processing or in sort of full text indexing. So if anybody wants to be involved, please let me know. Another area that came up in the panel in particular, and I think is critically important, is education. So we've actually focused a lot of our efforts on what could broadly be called education activities related to microbiology of the built environment, and I'm going to give you just a couple of examples of this. So one involves trying to educate and train people in the field, people that we already know are interested in microbiology of the built environment or in related fields, how to make the most out of data, for example, or how to better use web resources or computational resources. So for example, there's a really nice training operation called Software Carpentry that's now run by the Mozilla Science Foundation, and we've run, Jenna Lang, who's in the back somewhere, I think, from my lab, and Tracy Teal from Michigan State have run a couple of workshops related to this software carpentry activity, basically teaching scientists, engineers, mathematicians how to make better use of computational resources. Titus Brown just sent this to me a few minutes ago, saying that now Software Carpentry is starting a data carpentry workshop, which is going to be critical in the future because, of course, data, as we just heard in the panel, data sciences and manipulating and using and sharing data is going to be critical for all scientists in the future. And again, this is important for everybody. We would like the field of microbiology of the built environment to be in front in this area, to be expert at sharing, expert at using data, expert at everything that they have to do to become interdisciplinary future scientists. There are other workshops we've run. For example, CHIME is a software package that a lot of people use in the field to analyze microbial diversity data. So we've supported the CHIME group. We don't run the workshops ourselves. We've supported the CHIME group to run the workshop. That's sort of training activities. Another thing that I think is critically important, and this actually, we were interested in this But this came up in particular in the Sloan Foundation has an annual meeting of the Microbiology of the Built Environment program. And that, I think it was the first annual meeting of this program, we got suggestions from multiple people in the audience that engaging undergraduates more actively in research in microbiology of the built environment would be critical for the future of the field and would be very beneficial to us in terms of communicating about the field. And we knew, I know of lots of examples of this. There are some great examples out there, like the Malcolm Campbell has this great one at Davidson College, and there's this great phage program by Graham Hatfill, and Joe Handelsman and Tiffany Sang have this really neat thing called the Small World Initiative, which is trying to get people to grow up microbes from different environments and screen for antibiotic resistance from across the globe, basically to try and understand the diversity of antibiotic resistance mechanisms. I assume Tiffany is going to keep doing this. Joe now has moved to work for that Obama guy, so I'm not sure what exactly is going to happen to this program, but it's a really brilliant idea. So we've sort of taken this, what other people have done, and said, can we do the same thing for microbiology of the built environment? And David Coyle, in my lab in particular, has been working on multiple undergraduate research initiatives that tie into microbiology of the built environment. So the first one we did relates to what are called reference genomes. So if we want to analyze metagenomic data, it helps to have reference genomes from organisms that grow from that same environment. So we've been having undergraduate students go around to the built environment, culture organisms, screen them with ribosomal RNA sequencing to see if they are novel compared to what genomes are available. And if they are novel, we sequence the genomes with Illumina. The students have sort of learned how to do simple genome assembly activities, and then we publish very simple data papers that makes the data available to the community immediately. It describes how the data was generated, and it's part of this sort of future of data sciences where we're trying to push out data as quickly as possible and as openly as possible, but also help the field by generating genomes of organisms that were from the built environment that we can then try and interpret sequence data of samples, swabs, et cetera, from similar environments. And we've had, it's been great, undergraduates get a little genome report publication. They're not, the most groundbreaking papers in the history of the planet, but people are very happy to get these publications out. As part of this project, we realized that it was not straightforward to know exactly how to go from swabbing, a yoga mat, for example, or swabbing a toilet lid, or collecting something from the sewer system, I guess, and culturing an organism, typing it, identifying if it's novel, sequencing the genome, assembling it, and publishing the paper. And this undergraduate, Madison Dunitz, who's now working as a tech in my lab after graduating, has basically developed a full protocol. This is her presenting a poster on it, a full protocol for doing this that includes all of the scripts that are needed for doing this, every single instruction for going from swab to a paper at the end, including depositing the data. And this, the description of this should be submitted any day. We have another project I won't tell you a lot about that is also run by David Coyle, in particular in my lab, and Jenna Lang, and visiting student Matt Haggerty also were involved in coordinating this project. And it's also involved undergraduates doing research, but it was now using culture-independent DNA-based studies to sample the aquaria that we have in our biological undergraduate education labs and sampling them over time and trying to do something that is critically important for studies of the built environment, which is to record as much as possible all of the metadata associated with samples. So we're not just collecting things and sequencing them. They have been collecting about 20 chemical parameters. Paula, I don't know any chemistry, so I won't be able to tell you. Yeah, there are other people who do. They guided them on this project. We had a lot of help from Josh Neufeld, who this idea sort of came from, and Laura Sauter, who's in his lab. And they basically tracked the colonization of these aquaria over time and recorded nitrogen levels, pH, 10 or so other chemical parameters about the water samples and everything they could. And they're doing now ribosomal RNA sequencing and characterizing them and writing up group paper on this project. And this leads into the reason we got interested in this project was our activities related to what you can call citizen microbiology. And so I'll tell you about this in a second, but I just wanted to mention that there's this really interesting project that Josh Neufeld at Waterloo and this student in his lab, Laura Sauter, were involved in, which is getting people to send in their filters from their aquaria to try and isolate new organisms. And it seemed like this perfect mix of microbiology, the built environment, and citizen science and public engagement, because if you don't know, people who have fish aquaria are obsessed with their fish tanks, and they will do anything. They will record every chemical parameter you've ever imagined. They will send you samples. It's like where, I think Joe DeRisi left, but it's like getting the snakes in your mailbox. You could get aquaria samples for the rest of your life if you engage this community. And so this, in particular, was really sort of exciting to us because we think that engaging the public, not just engaging, so training the scientists is important. Engaging undergraduates in research activities is important. We want to engage the public and actually engage them in the research itself. And that's what, in the best case scenario, citizen science is about. And I got interested in this in particular about 4 years ago now at a meeting called Science Online in North Carolina. And there was a presentation coordinated by Karen Cooper, who does bird stuff at the Cornell Bird Observatory, and Darlene Cavalier, who runs a group called SciStarter, which is a clearinghouse of information about citizen science. And I was at this workshop, and I went up to them afterwards because she runs this clearinghouse, and I was like, well, why didn't you mention any microbial projects? Where are the citizen microbiology projects? I mean, who cares about birds and butterflies? And they're just not important. She said, well, actually, come sit there. She sat me down, showed me their website. She said there actually are a few. There's this water monitoring project where people are collecting water samples and trying to screen for various microbes. And so I thought this was just an amazing opportunity area. So we organized a workshop at UC Davis. Darlene was the keynote speaker at this workshop and was completely brilliant at showing people about how to do citizen science. We then ran a session at the American Society for Microbiology meeting on citizen microbiology, and we invited basically all the examples we could find of people doing citizen microbiology in some way. And it turns out there were a few. We actually started one ourselves in collaboration, where did Jack go? In collaboration with Jack Gilbert's lab, and a student of his, Georgia Bargil, has written up a bunch of this and analyzed data where we've been sampling microbes from phones from a variety of environments. There's this great, this was already mentioned already, there's this amazing Wildlife of Your Home project. Actually, Holly Menninger was at our workshop and was also at that workshop at Science Online. Rob Dunn and Holly together coordinate this amazing Wildlife of Your Home project where they're also looking at microbes in homes. That's me. My then 7-year-old daughter took a picture of me swabbing our front door as part of the Wildlife of Your Home project. And it turns out we weren't alone in thinking that this was an interesting area. It turns out there are many other activities that you could call crowdsourcing or citizen microbiology. Another one run by Jack Gilbert called the Earth Microbiome Project. There are a couple of human microbiome projects that are sort of more crowdsourcing than citizen science, and many other activities where people are now trying to engage the public in at least sampling microbes and hopefully in analyzing the data, in thinking about microbial diversity and that not all germs are bad. And we have this one project that came from this. So Darlene emailed me one day and said there's this call for proposals from Space Florida about doing projects on the space station. Can you think of anything to do with microbes? And I was immediately, I said, I want to sample the space station. I want to sample the space station. And she said, yeah, that's great, but that's not enough. You're never going to get funded if that's all you say you want to do. So eventually we came up through discussions with many people in this project, Darlene in particular. Darlene also runs a group called Science Cheerleaders. And that Science Cheerleaders group is a collection of scientists and engineers who are either ex or current NBA and NFL cheerleaders who do outreach related to science education. Darlene was a cheerleader, I think, for the Philadelphia Eagles before doing this. And this is an unbelievable group of people in terms of their commitment to science, their activity in science outreach, and their, breaking stereotypes about who can and should do science. And we eventually came up with this joint project with the Science Cheerleaders group to sample microbes from all sorts of different events and sites around the country. This is a map of some of the regions that we've sampled. That's one of the Science Cheerleaders sampling Buzz Aldrin's shoe at a Uri Night activity. A grad student who turns out who worked in my building is part of the Science Cheerleaders group, a cheerleader now for the Oakland Raiders, and a biomedical engineering graduate student that works one floor up from us. And we have this project where we've been collecting microbes from these different sites. We now have, a couple of weeks ago, there was a launch, finally, of the rocket containing our project. I don't think I have the video on here, via a Dragon X launch that sent this collection of microbes, 48 different microbes isolated from these different environments, up to the space station for, in theory, a NCAA tournament-style competition of growth on the space station. And the astronauts, Jenna Lang just told me that the astronauts did swabbing in the last few days, and we will be getting swabs back of samples of the space station. If you want to engage the public in microbial studies of the built environment, what's friggin' cooler than the space station? Maybe you could come up with things that, Mars, well, that's the next, sure. We'll be there. Anyway, so this has been this amazing project to me where it's got a lot of moving parts. It's somewhat complicated, but working with this group, the Science Cheerleaders, who have gone out and reached tens of thousands of people that we never would have interacted with otherwise in this area, and taught them about the hidden world of microbes that are on, the floors of basketball arenas or on people's shoes or on the space station, has been pretty amazing to us. And I think that these sort of education and outreach projects are really important. And this is not now directly related to microbiology of the built environment, but I thought I'd just give a plug that getting crowdsourced samples of the microbes from across the planet is a perfect part of what is my real dream and obsession, which is I'm a recovering birder, and I do like birds. I'm sorry I dissed them a few minutes ago. But I want a field guide to the microbes. And I want global samples and biogeography of microbes from across the planet. Forget New York. I want everything. I want the entire global planet and to understand microbes. And even though this is kind of absurd, it reaches the public in a way that many of the discussions that we've had about sampling microbes don't. They understand what field guides are. Many people have used field guides for different organisms. And talking about a field guide to microbes is one way to sort of engage the public more broadly in understanding microbial diversity. And the last thing I want to mention, I've hinted at already, and I just want to sort of put in a few plugs for it, is we think that for this field to be successful, for this field to be most successful, and to accelerate most rapidly, we need to embed a culture of openness and sharing in the field, even more so than has happened in some other fields. Many scientists love to share their information with each other, with the public, but there have been impediments, a variety of them, related to sharing. So one thing that we've been doing related to all of our activities is trying to teach what the benefits are to people in this field being more open. So that includes things like open access publishing, putting papers in preprint servers before you submit them to a journal. That's common practice in physics and math, not common practice in microbial ecology and microbiology. Even more open release of data. That's certainly happening, as we heard earlier, in some of the human genome-related areas. It doesn't always happen in every one of the microbial areas, but it could happen even more. Metadata about the data is critical. Just releasing the data is not enough. You need much more rich information about what that data is, and so on. I just thought, give a scientist a tool and you feed science for a day. Teach a scientist to be open and you feed science for a lifetime. trying to be what, I forget who that, the fish quote came from. So our students in all these undergraduate projects are publishing data papers as quickly as they get data. They're depositing the data in open data archives, publishing papers related to them. We encourage everybody associated with our meetings to post their slides to shared places like SlideShare. I posted my slides before my talk, by the way, so they're there up on SlideShare. We've written instructions for how to do this. We've made little video instructions if you don't want written instructions. One thing that we're trying to encourage is that if you share all this information, it would be nice to get credit for all your sharing. There's a movement called Altmetrics that is trying to improve our ability to track usage of papers, downloads of papers, paper-specific citations, downloads of data, data-specific citations, etc. There's even a manifesto related to Altmetrics. There's some really good blog posts by Heather Piwawar, who's one of the co-founders of a group called Impact Story. And I went to Impact Story and I uploaded all the things associated with microBEnet into Impact Story so you can track the usage of all the material. The most viewed publication is actually one by Jack Gilbert that I'm a co-author on this report from a meeting. The second most viewed is one on social media. So Holly Bick and Miriam Goldstein, who doesn't work on our project but is a collaborator of Holly Bick, wrote a paper on an introduction to social media for scientists. It's been viewed something like 100,000 times in PLOS Biology, and Holly Bick does training activities related to social media. She posts all the slides on SlideShare. So we're trying to figure out ways to use openness and to teach about openness at the same time. And that, I think, will accelerate progress in the field. It will help deal with information overload. It will help spread the news about meetings and workshops. It will help get people to know more quickly what type of education activities they can do. It will engage the public more broadly. And I think by doing these types of activities, we can really help this growing, interesting field accelerate more rapidly. There are many areas that we're hoping to do in the future. I think in the interest of time, I won't go into all of them, but we're trying to explore many different areas of sort of digital media. Again, going back to Paula's comments from the panel discussion, a lot of these ideas actually came from outside people. They came from reviewers of our proposals. They came from members of the Sloan Microbiology, the Built Environment community. They came from people. We've put out requests on Twitter and in blogs for, hey, have any ideas for what we should work on? Please give us suggestions. We're really open and interested in that. And I think I will end there and just thank all the people at UC Davis who've been involved in this project. It's been an amazing team at UC Davis, Hal Levin at the Building Ecology Research Group, of course, the Sloan Foundation for funding this work, and all of our collaborators in all of these different projects, many of whom I've mentioned already. And thank you for listening.
Do you have questions?
Yeah. Oh.
Yes, please.
I'm a physicist. Sorry.